All right, thank you everybody for coming to today's seminar on this cold January day. For those of you that don't know me, I'm Scott Dame. I'm here in my capacity as the director of the Targets of Cancer T32 training program, which is currently in its 51st year of support for postdoctoral and predoctoral trainees at our university. And I'm very pleased to present two of our very accomplished postdoctoral trainees who recently completed training on the T32. So we're going to have two 30-ish minute presentations. We'll do Q&A after each of them. Jason Jones will be our first presenter. So Jason completed his Bachelor's of Science training here at the University of Minnesota, as well as his PhD training at the University of Minnesota with Dr. Lipscomb. He then did a very short postdoc at St. Jude prior to coming back to the University of Minnesota to do his actual formal postdoc in the lab of Nick Levinson. And so Jason rotated off of the T32 just last year, and I'm happy to report that he was successful in securing a American Cancer Society Postdoctoral Fellowship Award so that he can continue the work that he's going to be telling you about today. So Jason, the floor is yours.
All right, hi. Today I'll be talking about how MYC protein stability is dysregulated in some human cancers and how aurora kinase A perturbs this process at the level of ubiquitination. And I'll be presenting some of our lab's collaborative efforts today. So I'll be sharing data from Dr. Nidhi Joshi. Whoops, let me put the laser up here. Postdoc in Nick's lab, as well as Dr. Katie. I'll be sharing data to help tell this story of how c-MYC is stabilized by aurora kinase A. So MYC is one of the most frequently dysregulated oncogenes in human cancer. And multiple mechanisms drive this oncogenic activity, like genetic aberrations, such as chromosomal translocations or gene amplification, both of which result in increased MYC messenger RNA and protein levels. And it's this excess of MYC that's exacerbating its oncogenic activity in cells. Another mechanism that can go awry is the signaling pathways that control the transcriptional regulation of the MYC protein, ultimately resulting in increased MYC RNA levels and protein levels. And a third mechanism is the stabilization of MYC. So typically here, if amino acid residue 58 is phosphorylated, so threonine 58, that is typically the signal for degradation, and MYC will get degraded in the cell. Whereas under conditions where if only serine 62 is phosphorylated, this is considered a stabilizing post-translational modification, and MYC can continue its transcriptional activity. In the Levinson lab, we are very, very interested in studying the biochemical and structural mechanisms that regulate MYC stabilization MYC stabilization. So here's two good examples of cancers where it's been clearly established that MYC degradation is dysregulated. So in neuroblastoma, typically what drives that is the amplification of NMYC. Whereas in hepatocellular carcinoma, more late stage hepatocellular carcinoma, the levels of c-MYC are elevated. Now, despite the two distinct origins of these two cancers, they do share one thing in common, and that is that proteasomal degradation is inhibited. So MYC proteasomal degradation is inhibited. And this is what we're very interested in studying biochemically and structurally. Yet to gain a better understanding of how MYC is dysregulated, we have to get a grasp on how is MYC normally regulated for degradation. So in humans, MYC has three isoforms. L-MYC, typically found in the lungs, or expressed in the lung cells rather. N-MYC, typically expressed in neuronal cells, and C-MYC, typically expressed in proliferating cells throughout our whole body. And when you do a protein sequence alignment, and what comes of that alignment analysis is that there's these conserved regions that over the years have been shown to be very important that regulate transcriptional activity as well as MYC degradation. So this is just a very simple diagram, just kind of outlaying how those conserved regions, where they are in relation to one another. Starting from the N terminus to the C terminus of MYC, we have this basic helix loop helix leucine zipper domain, and this domain interacts with DNA. And while it's interacting with DNA, these other conserved regions, known as MYC homology boxes, some of these serve as sites that recruit other proteins and enzymes that help regulate transcriptional activity. And importantly, MYC homology box 1 is what regulates the degradation of MYC. It contains a phosphodegron, and a phosphodegron is an amino acid sequence that, when post-translationally modified by phosphorylation, this is the signal for degradation, for MYC degradation. So in MYC, serine 62 is first phosphorylated. Then, and only then, does threonine 58 get phosphorylated. Once that event occurs, this is the signal to recruit an E3 ubiquitin ligase. And what a ubiquitin ligase does in this case is it post-translationally modifies MYC with a chain of ubiquitins. This chain of ubiquitins is the signal for proteasomal degradation. Now, in neuroblastoma and hepatocellular carcinoma, the two examples that I gave, this process is disrupted. Something is going on. So what's going on? Well, it was found in 2009 by Otto et al. In a paper that aurora kinase A stabilizes MYC independent of its catalytic activity. So Otto et al. In 2009, while studying neuroblastoma cell line where NMYC amplification is up, they found that NMYC is getting phosphorylated first on serine 62 by the CDK1 cyclin B complex, and then threonine 58 is phosphorylated by GSK3. So the signal for degradation is there. Additionally, all the machinery, all the biological machinery needed to degrade MYC is present in these cells. Yet under molecular conditions where aurora kinase A is abundant and at elevated levels, what they found out is that aurora kinase A and NMYC form a protein-protein interaction. And it's this protein-protein interaction that's stabilizing MYC. These authors also treated the cells with a ATP competitive allosteric inhibitor that binds to aurora kinase A. And then they found that some of these allosteric inhibitors disrupt this complex. So it breaks that protein-protein interaction. And what happens is those cells start to die afterwards. So these cells kind of become, I guess, dependent on MYC in a way. And so it's an actionable target. Fast forward to 2025, here at the University of Minnesota, Daniel Harkai's lab developed a degrader molecule, HLB, and they showed in MYC amplified neuroblastoma cells that when they used their degrader molecule, aurora kinase A was being degraded. And then in parallel, the MYC and MYC levels were being degraded at the same time. So this just highlights that whether you use a small molecule allosteric inhibitor for aurora kinase A, or if you degrade aurora kinase A, you alleviate this kind of mechanism that's stabilizing NMYC. Also in 2016, Dausch et al. Looking at hepatocellular carcinoma and then using mouse models, they also found similar findings. What was seen in neuroblastoma is that instead of NMYC, it's CMYC, and that aurora kinase A will form a protein-protein interaction with CMYC, and then that protein-protein interaction is stabilizing CMYC, allowing it to continue its oncogenic activity. And then using mouse models for their hepatocellular carcinoma, they treated it again. This is a allosteric inhibitor of aurora kinase A, and found that allosteric inhibitor had the same effect. It dissociated the CMYC aurora A complex. And I'm not an expert at reading these survivals, but this is a really easy one for me to read because it seems very dramatic, those results. And especially kind of qualitatively for me, you have the mouse that wasn't treated with the allosteric inhibitor, whereas the one was treated. It's a very clear contrast in what happened there. And then they also, kind of doing Western blotting can show that the part of the reason for this kind of survival was the degradation of MYC. Now, despite all these wonderful results, there still exists no FDA approved targeted therapeutic to tackle this problem. And kind of, I think my philosophy is, if we as a scientific community want to make better targeted therapeutics, well, we have to understand the biochemical and structural mechanisms that are regulating these processes, because at the end of the day, we're trying to develop molecules or other proteins that are targeting things at a molecular level. So we really need that fine-tuned understanding. Oh, wait, yes. Oh, so it does beg, so what is it about this interaction that's causing this inhibition? So as I mentioned before, so Dr. Nidhi Joshi, a postdoc in the Levinson lab, she recently published a paper where she used nuclear magnetic resonance spectroscopy, or NMR, to study the interaction between the N-terminus of c-Myc and aurora kinase A. And NMR is a wonderful method for studying these molecular interactions. So Nidhi, instead of studying the whole complex, she's looking at the N-terminus that includes the very important phosphodegron. And now what I'm showing here on the screen, this is a chemical shift perturbation plot of the N-terminus of c-Myc bound to aurora kinase A. And each of those bars just represents how strongly each of the residues is affected by aurora kinase A binding. And what I want to point out is that this plot shows that there's chemical perturbation shifts kind of spread out throughout this N-terminus of c-Myc, these broad perturbations. This just indicates that aurora kinase A is likely interacting with these different pieces of Myc. So this is a very extensive dynamic protein-protein interaction that's been classified in biochemically. And then importantly here, we notice this is the phosphodegron. That's the site where the E3 ubiquitin ligase has to recognize the substrate. And there's a huge chain. So aurora A has a huge influence on specifically phosphorylated threonine 58. And I also make note that residues flanking this phosphodegron are likely interacting with aurora kinase A too. Now, how? So we see that this reaction is happening, but what is it doing? How is it like inhibiting the degradation of Myc? And I think to get a better understanding at that, we have to look at the details of how Myc is typically marked for degradation. And that's by a E1, E2, E3 ubiquitin enzyme cascade that marks Myc for degradation. So just to walk y'all through this. So first you have this E1 ubiquitin conjugate. It will then transfer this ubiquitin over to an E2 enzyme to form this E2 ubiquitin conjugate. And this is known as the priming E2. It's the E2 that's going to post-translationally modify Myc with that first ubiquitin. Then that E2 ubiquitin conjugate will interact with an E3 enzyme that has Myc bound. And then the ubiquitin will get transferred from that E2 over to Myc. And in our studies, we're studying the particular E3 ligase that we're studying is SCF-FBW7. So after the E2 transfers that ubiquitin over, the E2 will dissociate off the E3, and then a chain elongation E2 will come in, and known for Myc, and is the name of that chain, one of the names of that chain elongation E2 is CDC34. And then through multiple rounds of ubiquitination, what happens is this K48 linked ubiquitin chain is built on the c-Myc onto c-Myc. And this is the signal for proteasomal degradation, specifically K48 linked ubiquitination. That just means that the ubiquitins, when they're added in the chain, it's linking one ubiquitin to the lysine 48 of the other ubiquitin. Now, where does aurora A interfere with this? So we know that aurora A forms a complex with c-Myc, but where is it interfering? If it's interfering with this process of degradation, where? Well, hypothesis number one is that aurora kinase A could possibly be interfering with how the substrate, how c-Myc interacts with the ligase. Hypothesis number two, does aurora A could interfere with the priming event? Because once it's primed, then it's chain elongation. Hypothesis number three, aurora kinase A is interfering with the chain elongation process. And so recent efforts by Dr. Katie Dunleavy, a postdoc in the Levinson lab, she found out that using biochemical studies that aurora kinase A disrupts K48 linked chain elongation. Yet importantly, priming remained intact, but K48 linkage is greatly impaired. And how she does this is she does an in vitro on the bench top in a test tube using heterologously expressed proteins that have been purified. And she does a time resolved ubiquitination assay. And this is an SDS page gel of that time course. Looking here at time point 0, this band right here represents unubiquitinated MIC, okay? And that signal, the signal, that black signal is coming from a fluorescent probe that Katie covalently linked to CMYC, and it's Alexa 48. So that's the signal that you see in this gel. And then as we go from left to right, just following this unubiquitinated, you see a decrease in the intensity of unubiquitinated MIC. Then looking at the 2.5 kind of minute time point, up here you see a band start to form, and then that band is increasing in intensity and also decreasing. What this band is that's the priming event. That's ubiquitin putting on the CMYC. That increases the molecular weight of CMYC, so how it changes how it migrates through the gel. And then a similar pattern, you see ubiquitination 2 event right here, increasing. So as these ubiquitins that are getting pulled on, the chain elongation is happening very rapidly. And at the end, this is this smearing pattern, as it's called, at 90 minutes. This is the ideal. This is chain elongation. This is polyubiquitination of the substrate. So what happens when we add aurora kinase A? To get right to the point, to look over here, K48 chain elongation is greatly disrupted. It's not completely gone, but that's a night and day difference. And then importantly, if we look at the priming's still intact. So where does that, Katie kind of ruled out some of my hypotheses based on her work thus far. So I can rule out hypothesis number 1 because CMYC is obviously binding to the E3 because CMYC is getting ubiquitinated. I can rule out hypothesis number 2 because I think I showed that priming is intact, although chain elongation is disrupted. So that leads me to this area is how exactly is aurora kinase A disrupting K48 chain elongation? I want to know where it's binding, what it's interacting with, and how it's causing these issues. And in order to do that, we need to visualize what's going on, and by gaining a structure perhaps. And so what I'm doing is I'm using cryogenic electron microscopy to try to resolve that architecture, to try to gain that complex, so we know exactly where aurora A is binding and what it's interacting with. And the whole cryo-EM workflow, it all starts with the biochemistry, good biochemistry, nice pure protein. So I heterologously express and purify all of the protein components needed to reconstitute this system in a test tube, and then characterize it biochemically to make sure it's active and everything's proper. And before I put my sample in the microscope, I have to make sure all of the subunits are there. I have to know that what I'm putting onto the microscope is the complex that I wish to look for. And to do that, I can use size exclusion chromatography. So size exclusion chromatography is a method that us biochemists use to separate molecules based on size. So in this chromatogram that I'm showing, the large SCF FBW7 CMYC Aurora A complex comes out earlier than smaller, more kind of compositionally heterogeneous complexes that come out. And so this peak right here represents my sample. That's all. That's my complex. So how do I know? Well, I can take this peak and I can run a SDS page that's Kumasi stained. And without going through all of these bands, just trust me, all of the components are there, okay? But very importantly, aurora kinase A is there, because that's the target, because this is the mechanism we're trying to get into. So now I have my sample. So then what we do is I take that sample, I put it onto a very small electron microscopy grid, and then using this instrument on screen right here, it's called a Vitrobot. What I do is I rapidly plunge freeze the sample into liquid ethane, which vitrifies it. So, and vitrification just means you make non-crystalline ice. And then I take that sample, put it in the microscope, and we take tens of thousands of images. Here's a representative image. In this image, I just want to do a plug because here at the U of M, we recently obtained a brand new cryo-electron microscope that's up and running. And so all of this cryo-EM work can be done here on campus right now. And this image is actually an image that was obtained from the U of M microscope. But this is what a typical image might look like. It's been enhanced a little bit for contrast purposes, but all those black minus there's like, this is junk, that's junk, that's junk, but everything else you see in there is protein. You're seeing proteins with your eyeballs in a 2D image, which I still think this is amazing that we can even see that. All right, so after I get all these images, then what we have to do is we have to pick all the particles in the tens of thousands of images that we have. Once we pick those particles, we kind of computationally cut them out. And then once we cut them out, we lay them on the table and then we align like particles with like particles. So matching shapes with shapes in order to build the signal up, because cryo-EM is a very low signal to noise method. And what that process is called 2D classification. And so these images that I'm showing you are 2D class averages that are a result of that 2D classification process. And what you're seeing two images of the same protein, just at different orientations. Because when I vitrify the sample using that Vitribot, you got to think those molecules are diffusing just randomly, and then I'm rapidly freezing them and trapping them in all different types of orientations. And then the next step, so this is where, much like a CT scan, where you need many 2D images in order to reconstruct a three-dimensional object, that's what we do too, using the central slice theorem. We can obtain through multiple steps of processing, we can obtain a three-dimensional model of our protein. Who cares? Why? You got a structure, big deal. Well, with these structures, this can drive hypothesis-driven biology. We can learn the architecture, how these proteins are, and then maybe develop new hypotheses to take and test into cells. Another thing that you can do is, medicinal chemists, computational chemists can use this information to possibly design new targeted therapies using information like this. So I proudly present, so to date, as far as I know, to the best of my knowledge, this is the first ever cryo-EM density map of an E3 ligase bound to c-Myc. So here in cyan, this represents, this density right here is c-Myc, which is the substrate. And do note, when the c-Myc protein that I used in this study was 150 amino acids long. I don't see all 150 amino acids in my cryo-EM density. I only see the phosphodegron and a couple residues flanking it. And we'll take a closer look at that in the next slide or two. But I just wanted to point that out. That means that the rest of the 130 so residues are very dynamic. Although it's bound to the E3, it's like a string waving in the wind possibly. And then the substrate recruitment enzyme, which binds the c-Myc substrate, is called FBW7, shown here in kind of pinkish purple. And then FBOX is interacting with this adapter protein called SCIP1. And SCIP1's function is to kind of link the substrate recruitment protein to the scaffolding protein called CUL1. And then here at the C terminal end of colon-1 is the E2 recruitment site where RBX1, it's a ring protein, is bound. And RBX1's function is to recruit E2 enzymes and position them for the chemistry. And so typically how this would work is you would have, for chain elongation, let's say that substrate gets primed, it has the ubiquitin on it, and then an E2, and then the chain elongation E2 would interact with that C terminus where RBX1 is, and it would transfer that ubiquitin onto the substrate and so on and so forth. But somehow aurora kinase A is interrupting this. And it's interesting because when we focus in on that substrate interaction, what I notice is I've modeled some of the residues into this density of c-Myc. So I've actually built atomic coordinates into here. The big thing that stands out is this really stresses the importance of 3-58 being phosphorylated, because ideally it really anchors this interaction down. To the kind of flanking that phosphorylated 3-58 there's a polyproline left-handed helix, which I feel like helps stabilize it too. But here is serine 62. It's not really bound. It's kind of flopping around in and of itself. So just to stress that. And even more importantly, to revisit the NMR data that we collected in lab, just want to draw your attention. So here, it showed in the absence of the E3 ligase, Aurora A interacts with MYC very extensively. And these interactions are at the phosphodieron and residues flanking the phosphodieron. But I don't see Aurora kinase A in my cryo-EM density map. So where is it? So my hypothesis is this, is that Aurora kinase A, under our current conditions with the sample that I showed you, it's dynamically tethered. And you could imagine a scenario like this where CMYC, the phosphodieron is bound to the FBW7 protein, but then Aurora kinase A is just it's dynamically bound to CMYC, like a balloon in the wind almost. Just kind of how I imagine it's going on. It's very non-canonical, but this might be happening. And so what would be needed, but then still, how is this, if this is true, this is a true scenario, how is Aurora kinase A disrupting K48 chain elongation? Well, we think it might be that when the chain elongation E2 comes and interacts with RBX1, that this might create a scenario where Aurora A can kind of wedge itself into this area right here. And perhaps that with that E2 there, it's enough like surface area for Aurora A to make a contact and kind of stabilize this interaction. It could also be that Aurora kinase A is just bumping into it, just a steric. Ooh, how much time? Okay, so that's that. And then just to show kind of how the dynamics of this molecule work, as I said, in this data set, the movie that I'm showing just shows all the individual movements that this molecule can accommodate to allow for K48 chain elongation. So in this scenario, you have all these movements in the F box region and the C terminal region, and that allows the chain to be built. You need this flexibility, this inherent flexibility in this molecule in order to do the chemistry, but somehow Aurora is messing with that. I could see how it wedged itself in there, and maybe it's perturbing the dynamics of that system. And so currently what I'm working on right now is putting just that sample onto the microscope where I add the chain elongation E2 to see if this stabilizes that complex. So in summary, I've shown you that MYC degradation can be deregulated in some human cancers, that it's a druggable target and it can help survival. The NMR data shows that CMYC and Aurora A have a direct interaction with one another. Biochemical evidence suggests that K48 linked chain elongation is disrupted. We still don't know where Aurora kinase A is, but the molecule is very dynamic, so we may need that chain elongation E2 in our sample to really figure out what's going on. And just want to thank everybody. Nick, Dr. Michael Sheedlow, before I came here, I had under a year worth of cryo-EM experience. And so Mike has been a great resource in that. T32, and then any questions there?
I've got a microphone for a question. We have time for one good question if you have one. All right. I don't know if it's a good question, but I'm just curious, has anyone else shown cryo-EM structures of Aurora kinase A? And I would think it's in your sample. So are you finding any evidence of it, like through the structures? None.
No, it's that, and that's what's challenging is like we were really expecting to see it because like the size exclusion data shows that it's there. Katie's done lots of work to show that it's there. It can all bind as one big complex. But again, to go back to the hypothesis is that I think it's just a very dynamic interaction. And cryo-EM is not a hammer that can solve all the problems, but so we might be missing something in our sample that's required to maybe stabilize it so that we can see how Aurora kinase A is disrupting that chain elongation. Hi.
So just really quickly follow up. And we need to have a microphone for your question. Yeah.
Sorry. So building off that, it's clear that the big complex is dominating your particle picking. Is there a way, maybe this is naive, but can you exclude the big complex, focus on aligning on anything else, and maybe you'll catch the other part of CMYC and Aurora in there already?
So maybe. The thing is right now I can't, in my data, because Aurora kinase A is so small, it's a very difficult target and it doesn't have a lot of features to it. It's just kind of like this little peanut, if you look at it from a big level view. The MYC thing is because it's so dynamic and it's literally a chain of amino acids and doesn't really have, as far as we know, there's really no secondary structure that can really, the electrons from the microscope have nothing to, the signal is just so bad. just so bad for that polypeptide chain. But the hope is that maybe something will stabilize some kind of structural change that we could actually observe in the cryo-EM. Thank you, guys.
All right, let's say thank you one more time, and we'll get switched over for Alex's presentation.
Okay, and so our next speaker is Alex Tsai. So it was wonderful having Alex as part of the T32 because he's a physician scientist and really kind of held our trainees' feet to the fire in terms of translational relevance of a lot of the projects and questions that we were discussing. Now, Alex is also a homegrown talent, got his Bachelor's of Science here at the University of Minnesota, then went to the University of Maryland to get his MD and PhD degrees. Came back to the University of Minnesota, and he was part of our physician scientist training program where he completed his residency and fellowship training and was a member of our T32. And then Alex rotated off of the T32 to accept a faculty position here in the Division of Hematology, Oncology, and Transplant in the Department of Medicine. So he's an assistant professor, and he'll be able to continue the work that he's going to tell us about today.
All right. Hopefully that looks okay.
Bring it.
Oh, the participants.
Yeah. There you go.
All right, thanks, Scott. So I'm going to tell you a little bit about T-cell therapies in the context of pancreatic cancer and beyond, actually, today. But I decided to gear my talk sort of through the lens of being a former T32 fellow. And I think that Scott and Carol before him would agree that two of the main goals of sort of the training grant are to provide opportunities for people to continue training and to learn lessons and to gain experiences from those, that training period. And then two, for me, of course, providing sort of a springboard for me in terms of transitioning towards independence. So I'm going to tell you kind of two lessons that allowed me to achieve both of those elements during my several years on the T32. So first, just very briefly, pancreatic cancer. I think all of you know that this is a really challenging disease. Even though it's just the 10th most common malignancy in the US, it accounts for the third most cancer-related deaths here. And that's due to a variety of reasons that I'm going to highlight a few of. First of all, half of our patients are diagnosed with metastatic incurable disease. And furthermore, these patients have terrible prognoses. In real-world studies, these patients survive on average just 6 months. And even if you're lucky enough to be able to undergo surgery after chemotherapy, 4 out of 5 patients who undergo chemotherapy or undergo surgery will have recurrent disease within the first couple of years after their surgery. And ultimately, that results in a roughly 13% cure rate for this disease. Part of the reason for that is our treatments really are limited. Okay, so here are 2 New England Journal clinical trials, both published in the early 2010s, and these form the standard therapies for pancreatic cancer today still. Meanwhile, during those same 15 years that have passed since, these are sort of a very brief and abbreviated lists of different immunotherapies and targeted therapies that have been approved for various cancers. But unfortunately, none of these are applicable to pancreatic cancer. And part of the reason that at least these immunotherapies are ineffective is that this disease is considered a so-called quote unquote immunologically cold disease. And here are a bunch of different either IF or IHC images that I've pulled showing the density of CD8 T cells within tumors. And just, broadly, you can tell that these cells, which we really believe are one of the core anti-tumor T cells or immune subsets, are really sparse in this disease. So moving on to sort of this first lesson, learning from your challenges. Okay. So if T cells are so limited endogenously in pancreatic cancer, well, maybe there's a method where we can sort of overcome this exogenously. And one way that's been developed to try to achieve this is adoptive cell therapy. This is actually a very old approach. It's now about 40 years old. And it's relatively simple in that you take T cells out of a patient, then you genetically engineer them to express some sort of receptor that makes all of these T cells somewhat homogeneous and all target a tumor. And then you reinfuse those engineered T cells back into the patient. Okay. And so what we've done is we've adapted this to our mouse models. So we implant a pancreatic tumor into the mouse. We use cyclophosphamide to lymphodeplete this mouse host to provide sort of an immunological space for our T cells to engraft and expand. Then we performed adoptive cell therapy. We might provide some additional therapies, and we look at outcomes. Now, to study this in mice, you need sort of a model. And so here's just a very brief schematic of our model that we study quite a bit. We know that mesothelin represents a good target on pancreatic cancer cells. In humans, about 90% of patients overexpress this protein. And we target that mesothelin protein using a special TCR that we call the 1045 TCR. We know that if we just transfer these cells that express the 1045 TCR, that they provide limited efficacy. And one of the reasons that we hypothesize for this is lots of immunosuppressive molecules that are produced by either the tumor or immune cells and other cells that are co-opted inside the tumor. And one of the primary cytokines that does this is TGF-beta. It's very abundant in pancreatic cancer. And so to target this, we knocked out TGF-beta receptor 2, which ultimately allows for these T cells to become sort of impervious to suppressive TGF-beta signaling. And so it took a mouse we needed to generate a mouse. And that process, you can see that there were 5 alleles to cross here. It took about a year. And so we were very excited when we first bred this mouse. We used this mouse to create T cell donor cells. And then we put it into our adoptive cell therapy model. So we implanted so-called KPC2 pancreatic tumors into the pancreas of mice. We did adoptive cell therapy, and then we looked at tumor weights about 10 days after transfer. And what we saw was either with TGF-beta receptor 2 wild type T cells or with knockout T cells, there was maybe some hint of efficacy, but there was a problem, there was a challenge. And that was that, even in the knockout T cells, we didn't see any sort of difference between those two groups. So we wondered if there was a problem with our T cells. So just briefly, I'm going to take you along one more slide of background and tell you about how T cells differentiate within tumors. We and others feel that these cells probably begin their lives as so-called T-stem or stem-like T cells. These, like all stem-like subsets, have self-renewal properties. And upon initial antigen exposure, they become T-pecs or progenitor exhausted cells that upregulate this molecule that many of you have heard of, PD-1. And upon further antigen exposure, they become effector cells. These are sort of the soldiers, the workhorses that are really going to do the anti-tumor work of the immune system. And as they continue to see chronic antigen, they become so-called exhausted, a phenomenon where these cells lose function and lose proliferative capacity. And so I wondered if this sort of typical canonical T cell differentiation was occurring in our model. And just of note, our typical immunotherapies, checkpoint blockade therapies, are thought now to target this T-pecs precursor cell. So here we're looking at that stem-like population. I've gated on them in the non-draining lymph node, the draining lymph node, the spleen, and the tumor of mice that underwent adoptive cell therapy. And you'll have to take my word for it, but you'll note that nearly all of the cells form T-stem or stem-like cells within the lymph nodes. And then for our model, very high percentages or frequencies of our transferred cells also form T-stem within the spleen and then the tumor. And then if we gated on these and then looked at PD-1 expression, therefore looking at those TPECs, what we saw was a lot of nothing. So these cells seem to remain fixed in this T-stem state without progressing on to this TPEC state. They never upregulated PD-1. So then we looked downstream to see if there were effectors and see if there was exhaustion forming. And again, we didn't see a lot. So while our TGF-beta receptor 2 knockout T cells did form more effectors, only about 10% of our cells really robustly form these effectors. And moreover, very strangely, really none of our cells were becoming exhausted. And that's a phenomenon that we routinely see in all of our models. And so all of this entire differentiation pathway seemed blocked. And so we hypothesized then that this differentiation is dependent on antigen. So we wondered whether or not antigen was insufficient in our system. But when we looked at these cells, when we looked at a variety of different KPC cells or pancreatic tumor cell lines, we saw robust mesothelin expression. And as you'll note, you need both that peptide antigen along with MHC class 1, and we could see plenty of MHC class 1 as well, so long as we stimulated our cancer cells with interferon gamma. So all of the components of the recipe seemed to be there, but there was one other potential factor, and that's that these proteins need to be processed into peptides and then loaded onto MHC class 1 in order to be ultimately presented. And so this is actually a complex pathway with lots of protein machinery, and any loss of any component of this sort of presentation pathway can disrupt ultimately the presentation of a certain target. So antigen expression doesn't necessarily mean or equal antigen presentation. And so what I did is I designed a way to sort of bypass that, a very simple approach. Basically, what we did is we engineered our cancer cells to overexpress just the peptide antigen, just this little fragment of the mesothelin protein, targeted that to the endoplasmic reticulum, which allowed for sort of passive loading onto MHC class 1. And then we took those cells and we re-implanted them into tumors, into mice, established tumors, and did adoptive cell therapy once again. And here we saw a sort of rescue of the system. We saw improved outcomes, slowed tumor growth with the knockout cells. We saw better engraftment and expansion. So those knockout cells also were much more prevalent in spleens and tumors. And we also formed we also saw that those cells readily formed these TPECs, or these progenitor exhausted cells. So we had sort of restored at least this first step in differentiation. Downstream, we saw also that these knockout cells form better effectors. So nearly 50% of our transferred T cells were forming effector T cells. Again, those sort of soldiers that are doing the anti-tumor work here. And at least there was a little bit of signal towards reduced exhaustion, but notably all of the cells started to show evidence of exhaustion. So by providing antigen, we had sort of rescued this canonical differentiation pathway and solved our sort of challenge. And finally, these knockout cells, which many have shown previously, when you block that TGF-beta signal, you get improved function. And that's what we saw here. So if we pulled T cells out of those tumors and then re-stimulated them, we saw much improved effector cytokine production, including interferon gamma and granzyme B. So the lesson here is to learn from our challenges, that these challenges are opportunities to show and learn new things. Here, we learned that antigen availability is probably a conserved barrier to adoptive cell therapy efficacy in pancreatic cancer. So with my remaining time, I'm going to talk about that second lesson, how to sort of leverage new observations and turn them into new projects that for me could catapult into sort of my first sort of independent project. And it centers on a completely different topic, and that's chemoimmunotherapy. This is a combination between chemotherapy and then our typical checkpoint blockade immunotherapy medications. And we're starting to use these a ton. So here's just an abbreviated list of all of the different cancers that we tend to use this type of combination therapy in the first line or a curative setting, okay? And so they're very prevalent now. Then there are limitations though. So first of all, only about 10%, if you average all of these clinical trials, 10% of patients benefit from the inclusion of immunotherapy. And secondly, we have no good ways of picking out those 10% of patients. So all patients will be exposed to potentially harmful side effects of immunotherapy, but only 1 in 10 will actually benefit from the inclusion of immunotherapy. And part of that, that lack of knowledge is because we've studied this purely in the clinical setting, purely in clinical trials. And we really haven't asked this question from a basic science kind of standpoint. And so the reason why I bring up this topic is because of this small observation. So what we were doing is we were trying to optimize this adoptive cell therapy protocol. We're using this cell line that allows us to track the endogenous tumor-specific cells. So we know exactly which T cells are actually specific for the tumor. And we're using this drug cyclophosphamide to sort of create that immunological space. And if I looked at the endogenous T cells, what happened is not a lot if you looked at bulk cells, okay? So this is number in the spleen, the draining lymph node and the tumor. There was some decrease in the tumor, but not a huge loss, maybe tenfold. But what happened if we looked at the tumor-specific T cells was much more dramatic. So in fact, these tumor-specific T cells, at least in the tumor, became difficult to actually even find. So cyclophosphamide in this case, which by the way, oftentimes serves as a chemotherapy agent, was depleting our tumor-specific cells. And that led to this question of whether or not this type of combination is actually rational, okay? And whether or not that chemotherapy was selectively killing our tumor-specific T cells. And so I tried to leverage our pancreatic cancer models to try to answer this question. And I used standard of care chemotherapy with our tumor cell lines. So gemcitabine plus or minus Abraxane here. So I just exposed our mice with tumors to 2 doses and then looked at tumor weight. And you can see that chemotherapy is effective in this model. No surprise there. And then I looked at the different immune subsets. So broadly, all immune cells were completely unchanged, both in the draining lymph node, the spleen, and importantly, the tumor as well. Myeloid cells were completely unchanged as well. CD4 T cells, completely unchanged by chemotherapy. But the story changed when you looked at CD8 T cells. So here in the tumor alone, you could see that there was maybe a 10-fold reduction by chemotherapy in the CD8 T cell population. And then if I split that population again into sort of a mixed population of so-called sort of bystander cells with maybe a few tumor-specific cells, but then also into a bona fide known tumor-specific CD8 T cell population, what I saw was a difference there. There was a small reduction in these bystander T cells within the tumor, but there was a much larger 10-fold loss of our tumor-specific T cells within the tumor. And furthermore, even though there was no change in the T cells in the draining lymph node and the spleen, so tissues away from the tumor, what we saw was a loss of those T cells throughout all tissues, the tumor-specific T cells, I should say. So then I returned to this sort of differentiation schema and asked a new question. Well, we know that these sort of soldiers, those early effector T cells are probably the most proliferative and they probably have the highest functional capacity. But because chemotherapies target highly proliferative cells, I asked the question of whether or not these T cells, this subset was particularly sensitive to chemotherapy. And here are the results. So if we honed in on just the TPEX, those early effectors, and then the late effectors, here's what you saw. In terms of number, everybody was damaged. Everybody was somewhat successful. But this was perhaps a 5-fold change, a 10-fold change in those late exhausted effectors, but there was a nearly 50-fold loss of these early effectors within tumors. So this cell subset seems to be the most susceptible to chemotherapy. So finally, I began introducing this topic by talking about chemoimmunotherapy, and then I showed you data from chemotherapy alone. So what happens when I incorporate immunotherapy into this treatment algorithm? So I treated mice exactly the same way, and then also incorporated in one cohort anti-PD-L1 or checkpoint blockade. In terms of efficacy, here's what we saw. All therapies were effective, chemotherapy alone, immunotherapy alone, and the combination. But importantly, chemoimmunotherapy, shown here in purple, had no additional benefit beyond either monotherapy. Okay. And then in terms of the CD8 T cell populations, again, any mouse exposed to chemotherapy is either in green or purple here. You can see that these two groups of mice really suffered losses specifically to their CD8 T cells, and that it appears to be a much larger loss in the tumor-specific T cell population as compared to the sort of tumor non-specific T cell population. And then if I separated again and looked again at those different subsets, the TPECs, the early effectors, and the late effectors, we saw the same effect. We saw a big loss in the early effectors, and importantly, immunotherapy did not act to sort of counteract that toxicity. So despite the inclusion of immunotherapy, we are losing all of the sort of active anti-tumor T cells within tumors and also within other tissues as well. So, that little observation, something that we weren't looking for at all, but happened to see, has turned into sort of a big project, and one that I think has big ramifications for lots of different people with lots of different types of cancer, all of which are treated with chemoimmunotherapy. So early days still in this project, and I have lots of additional goals. So first, you'll note that I did all of these studies in pancreatic cancer where we don't use chemoimmunotherapy. So the idea is to expand this type of study into other model systems, including lung cancer or other cancers that are more readily treated with chemoimmunotherapy. And then follow that with ways to try to protect those T cells. Well, if the chemotherapy is killing the T cells, then one idea to sort of rescue or improve the efficacy of chemoimmunotherapy is to protect those T cells. And so you can imagine lots of different approaches. Here I've noted a few of them that I won't go into to try to protect those T cells and preserve them in the face of potentially toxic chemotherapy. And then extending these types of studies into human T cells. There are lots of data sets out there that I can use, including potentially collecting new clinical samples as well. And then, of course, in the long term, the hope, of course, would be to try to develop some sort of clinical trial to improve the efficacy of these types of treatments. So that's all I have. Hopefully I finished on time. Happy to take any questions, and thanks for your attention.
Again, I've got microphones for anyone with a question.
Alex, that was really cool. Very nice work. Just out of curiosity, from your sort of towards the end, when you had the model, the KPC model, and the checkpoint blockade worked as well as chemo in the mice.
Yeah.
That doesn't translate, right? You don't
Correct.
see the same effect.
Yep. So the efficacy for immunotherapy in humans is about 5%, probably less. We've engineered that model to be an immunotherapy responsive model. Immunotherapy is not curative, even though it was pretty effective, you saw there, but those tumors will rebound very quickly and develop resistance. So it's sort of a model to study just broadly what happens when you have an immunotherapy responsive model in the setting of chemoimmunotherapy.
Yeah. So I think, the one of the points that comes up really clearly is sequencing. Right, and that's sequencing not as in genome sequencing, but drug sequencing.
Yep.
And I think this issue is a really hot topic in radioimmunotherapy and chemoimmunotherapy.
Yep.
And there's pros and cons, right? You activate the immune system, you hit it with chemo, and you wipe it out. You come in with chemo, you create a mess, and the immune system doesn't activate. Do you think so? In liquid tumors, there's really good examples that you can make chemoimmunotherapy work. Do you think in the solid space there's hope, or should we really be thinking about making immunotherapy more robust and combining it with surgery and sparing the radiation and chemo are trying to understand which patients get one or the other, but is it really futile to do both?
Yeah, I'll answer your question in a couple of ways. Number one is that we do have good examples from solid tumors that this type of combination works quite well. It's just it only improves outcomes in a small subset of patients. So we're using it every single day. This is probably the most common type of combination that we are using in solid tumors. And yeah, so the goal is to improve that number, right? To improve efficacy. In terms of sequencing, I'll answer your question with studies that we do know that aren't exactly the same, but in melanoma, for example, we have two core therapies. We have targeted therapies, and then we have immunotherapy. And both are quite effective, and we tried to ask the question through actually very good clinical trials of what happens, what should we start with first, right? And there are certain scenarios where it makes sense to start with one above the other, but there are many scenarios where we don't really know. And so we have sequenced those, and the consensus has been that starting with immunotherapy is probably more effective first, okay? And so that is not exactly the same sort of scenario, but I do think it lends even more fuel to this sort of question that I'm asking, because you have to wonder about what happens to the immune system after you go through targeted therapy and lots of other sorts of therapies. Is the immune system, can we not rescue the immune system with immunotherapy any longer?
All right, are there any more questions? Let's say, oh, hang on, one more question.
Okay, you mentioned what I was curious about, so I was like, I gotta ask. I was curious about the targeted therapy question. And then you mentioned melanoma, and I was like, oh yes. And so I was curious, like, when you have targeted therapies, do you think that it is possible to have some of them that are sparing of the immune cells, but then also a lot of those targeted therapies, like I immediately thought of melanoma and BRAF.
Yep.
And that's targeting still a proliferative pathway. And a lot of targeted therapies do target proteins that are involved in proliferation and antigen recognition. And if you think those would still be potentially harming the T cells, or if there's a way to kind of spare them, or if it would be more so on the side of the cancer cells getting destroyed in a way that makes them not targetable.
Yep. So that's a good question. And those are active, ongoing questions that we're trying to answer still. So we'll just take the MAPK pathway along with melanoma and actually pancreatic cancer. For example, we have really robust data from BRAF inhibitors for melanoma, and we still don't have a great sense of whether or not that's actually affecting T cell proliferation, because that MAPK pathway, RAF proteins participate in signal transduction for T cells as well. And we know that those V600E inhibitors that we use typically in melanoma, they're not very specific, okay? They target wild type BRAF as well. In pancreatic cancer, we have the same question. On one hand, we have a lot of data that says that targeting the MAPK pathway will improve sort of immunologic outcomes or measures in the cancer cell. Okay, so it improves antigen expression, improves MHC class I expression, those types of studies, right? But a remaining question still is whether or not, KRAS inhibition and RAS inhibition in pancreatic cancer cells is going to, or in T cells in pancreatic cancer is going to really harm T cell efficacy. And there's some preliminary studies coming out in pancreatic cancer, which aren't very robust. And it's certainly an active question in our lab too, is how to form better combination therapies when there's a risk of T cell toxicity. So, yeah.
All right, perfect. Let's say thank you to our T32 alumni, and thanks for a nice conversation.